So welcome everyone back, and just a reminder, I'm sure everybody's heard this a number of times, but we'll be using the Q&A feature for the questions, which you can post during the talk and you can upvote. We won't be using the chat or the raised hands for questions during the session. But the speaker has posted her slides already to the feed if you wanted to download the slides. Today we have first Andreella, who's a PhD student at the Department of Statistical Sciences at the University of Padua. She works on statistical methods that can improve the statistical analysis in neuroscience and connecting psychological and statistical methodology. She's worked on methods including spatial regularization of the Procrustes method and alignment of brain data from different subjects, and also in the framework, the All Resolutions Inference framework for brain imaging, which I think we're going to hear about today. She collaborates with colleagues at Dartmouth and Leiden University. So thanks so much for joining us.
Thanks for the introduction. Thanks everyone for being here. I will present the pARI package that stands for Permutation-based All Resolution Inference. That is a joint work with Professor Livio Finos from the University of Padua and Professor Jelle Goeman from the Leiden University Medical Center. This package permits to perform valid double dipping using this method that is called permutation-based All Resolution Inference. So, as you can imagine, we are in the multiple testing problem, and when we have this problem, the researcher needs to decide which type of correction used in order to handle potential false positive. And so the researcher can decide to correct a level of single variable or a set of variable. And now, and so we can have this trade-off between low power and low information that I will explain with a real example. I focus mainly in functional magnetic resonance imaging data. And so I will explain at first this method applied in this type of data, but we will see that this method is very general and that we can use it in a very different framework. And so we have fMRI data that are composed by scan taken over time, and each scan is composed by voxel, a three-dimensional voxel. Each voxel captures an indirect measure of brain activity, and so we have a test statistics for each voxel corresponding to the null hypothesis that the voxel is not active. One of the most used methods in order to analyze the brain activity in fMRI data is the cluster extent based thresholding. How it works? We have the test statistics, one for each voxel, so one p-value for each voxel. We put this p-value in a graph like this, so contiguously. And we have two thresholds. The first one constructs the cluster. So in this case, we have three clusters. And the second one say if the cluster is significant considering its size. This method is very used because it has more power respect to a method that corrects at the level of voxel, but has some problem. The first one is called the spatial specificity paradox, because if we reject the null hypothesis, we only know that this cluster is significant, but we don't know how many voxels are significant inside this cluster. So the larger the cluster, the weaker is the finding. And we cannot infer inside this cluster without falling into the so-called double dipping problem, also called a circular analysis or cherry picking. This problem was resolved by Rosenblatt and other authors, proposing the All Resolution Inference method that permitted to infer inside that cluster, so infer inside the data-driven cluster. This method makes inference on the number of truly active voxels inside that cluster. We can apply this method all the time that we want to infer inside a data-driven cluster, and also not data-driven cluster, every type of feature sets. So for example, also we apply it in the cluster AAG data analysis or gene expression cluster analysis. I will start with some theory in order to understand how this method works. We just say that we are in a multiple testing framework. So we have M feature to test, and so we have M hypothesis. We consider the set of true discovery unknown, called A, and the cluster of interest S. We want to make inference on the size of the intersection between these two clusters, between these two tests, the set of true discovery and the set of cluster. And so the All Resolution Inference is based on the closing testing procedure for controlling the family-wise error rate, and is based on the local test used inside this closing testing procedure. How works this closing testing procedure? It rejects any intersection if all intersection hypotheses that involves this intersection are rejected by a valid local test. So generally we need to test we need to perform a number of tests equal to the power of m. That is a very big problem in a computational point of view. But Goeman, in a very recent paper, finds a very good shortcut and using the definition of a critical vector. So I will define what is the critical vector. The critical vector is a vector with the length equal to the number of features that satisfy these inequalities. What it means that this critical vector cut the null distribution of the p-value in order to have below it the alpha percent of the distribution. Having this definition, we have this lemma that define the lower bound for the number of true discovery in this way. And this lower bound is valid for each type of S, so cluster, the set of features that we decided to analyze, because the inference is made simultaneously for all possible subset of hypotheses. So it satisfies this inequality. How works this computation? The computation for the lower bound of the number of true discovery. We have, so in this plot, we have the observed p-value and the critical vector. And we computed the distance between these two curves, and we take the maximum of across U of 1 minus U plus this distance. In this case, for example, we have the maximum here. So considering a set of 1,000 hypotheses, we have around 200 of, at least 200 of true discoveries. In the parametric version, so the one proposed by Rosenblatt and other author, the local test used is the one based on the SIMS test, defined in this way. So in order to be the closing testing procedure valid, we need to have a valid local test. And so the parametric All Resolution Inference assume the SIMS inequality. That means that we need to have a positive regression dependency on subset assumption. That is very common in other controlling procedure, like the false discovery rate proposed by Benjamini. However, if we have a very strong correlation between tests, between features, we can, the parametric All Resolution Inference can be conservative. For that, we propose a permutation approach of this method. And in order to construct the critical vector that we define here, we use in this part the permutation theory. So we compute the permutation p-value null distribution, and we define the family of candidate critical vector in this way. In our case, the critical vector depends on this parameter that we call lambda calibration parameter, that is very useful in order to calibrate our critical vectors. So in this figure, we have the null distribution of the p-value, and we have some candidate critical vector, and we choose the one, the dotted red one, that cut the null distribution in order to have below it the alpha percent of this distribution. So we propose a different type of family of candidate critical vector, the SIMS-based one, that is very similar to the parametric version, but we have the lambda calibration parameter instead of the alpha value. The one based on the asymptotical optical rejection curve, and the other that I will skip for lack of time. So the main result is that our critical vector satisfy the definition of critical vector that we just see here. And if we consider the Simon-based family and we put the lambda calibration equal to alpha, we have that the parametric All Resolution Inference proposed by Rosenblatt and the others is equal to the permutation-based one. And then after this part, we present some function that you can find in the pARI package. So you can find the package in my GitHub webpage, and we have two main functions: the pARI brain and the pARI. The pARI brain is constructed for the fMRI framework. And while the second one, you can use it in every type of framework. So in the pARI brain, we need to have the COPEs that are the contrast map, and in nifty format, one for each subject. You can specify the threshold in order to construct the cluster, and you must put the mask and the alpha value that you want to consider. In the same way, in the general pARI, you need to put the data, that is a matrix with dimension equal to the number of features times the number of observation, and the set of interest. This set of interest can be a vector of indices of hypothesis of interest, or a vector with length equal to the number of features, where different values indicate different sets, like the output from a cluster or kappa means and so on. And the type of test. Now we have the one sample t-test and the two sample t-test, but we want to extend it. And so we analyze some fMRI data, and we analyze the auditory data collected by Pernet and the other authors, where we have 114 subjects listening vocal and non-vocal sounds. So we analyze the contrast between vocal and non-vocal stimuli, and we flipping the sign of this 114 voxel Y contrast map. You can download the data using the fMRI data package that I put in my GitHub. And the function is very simple. We put the COPEs, so the contrast map, the cluster. So in this case, we put directly the cluster map. So you can decide to put or the threshold or every cluster that you want to analyze, and the mask and the alpha. This is the results. And so I put here only one cluster using a threshold equal to 3.2. And so in this cluster, we can see that at least 92% of voxel are active. So our true discoveries, and if we can also drill down inside this cluster, consider a higher threshold, and so in this case, if we consider this cluster, we have four other cluster, and we can also associate the lower bound for the number of true discovery for each of these subclusters. You can also create a true discovery proportion map using the function map TDP. So you can see the spatial location of our cluster and the associated true discovery proportion. Okay, so I also apply the method in some simple gene expression data analysis. And so I analyzed the data from Bottomley and et al. And in this case, we have 1,000 genes, around 1,000 genes and 21 samples with 2 types of strain. And we perform in this case a 2-sample t-test for each gene, and I consider as a set of interest the one computed by AK cluster. So we put the data, we put the clusters and the type of test, the label in order to perform the 2-sample t-test. And this are the results. So we have 4 clusters by AK cluster, and we can associate for each cluster the number of true discovery. So in this case, in the first cluster, for example, we have at least 27 genes truly active, truly significant. So concluding, the method is very flexible, mild, and postdoc, you can choose the cluster of interest as many times as you want, and the cluster can be data driven or not. In particular, it resolves the spatial specificity paradox of cluster fMRI data analysis, and is very general because we can specify different type of critical vector using the definition of families of confidence bound, and the permutation approach permits to take into account the dependent structure of the data and makes no assumption on the null distribution of the p-value, but we have obviously the exchangeability assumption because we are in the permutation framework. The power of the method does not depend on the number of features set that we want to test. So you can choose this features test as many times as you want, really, and still controlling the family-wise error rate. And so I wanted to thank the group that worked with me on the paper that is in archive now. Livio Finos, Jelle Goiman, Jess Hemmerich, Wouter Wieder, and thanks for your attention.
Thank you. So feel free to add questions in the Q&A session, and I'll start off. So I think this is an incredibly important area of method development, and I have a simple question maybe. So for an end user, so someone looking at the imaging data, or we have a lot of people here in genomics, you had the visualization at the cluster level. How have you thought about ways to visualize then also the testing within clusters? Is there a way to allow people to kind of go visualize across scale to see the results at different scale?
Yeah, it's possible, but I didn't develop this tool, but I think it's very important. There is now the Shiny app for the parametric version, but it's only that we can only use fMRI data. But it's very useful because the people can just put the cluster, the data that they want to analyze, the type of test, and iterate on the true discovery, the lower bound of the true discovery proportion, going everywhere in the brain. So, yeah, we need to do that, but yeah, there are some issue. some issue that we need to resolve, because being a permutation-based approach is slower than the parametric one, because we need to do the permutation. Yeah, so, but now takes around 3 minutes for 200,000 voxel and 1,000 permutation.
Yeah, that's what I was going to ask. I was going to ask about the sample size and the time. So it's that's not very much. I mean, so.
No, yeah, for the neuroscientist is good. I mean, 3 minutes for 200,000 voxel and 1,000 permutation is manageable. Yeah.
See, people are shy about asking questions this morning or this afternoon, where you are. So yeah, I mean, we were discussing before. I think this is also key for genomics, to allow people to kind of navigate at different scales and perform the inference at the level the data provides. That's also, I've been stealing a slide from Susan when I talk about this idea.
Okay.
The summarization slide.
Right. And well, it's the problem that we have a lot in immunology, where people use the same data to declare a cell type, and then they make summaries of the cell type, but then they make tests with regards to the cell type that they made with the data, which is just the same problem as you have. So when you do single cells, or you do any kind of immunology site-off, we tend to want to summarize very quickly into a cell type. So I was saying that in single cell and site-off, people do a lot of declaring the cell type. They use the same data. So they're using the same data, they're double dipping in that way. So that's why that.
Yeah, it's very famous, the double dipping problem in a lot of framework in neuroscience. The neuroscientists did that for a lot of years. I mean, I think only in, I don't know, I don't remember the year of the first paper that talk about circular analysis and double dipping, but it's very recent, I think.
There's a question from Aideen. Can this method be applied or extended to mammogram data?
I don't know really, but I think yeah, we can talk about that. Depends of the type of test and the type of hypothesis. So how the problem is constructed, and but the method can be applied in various situations, like not fMRI data. Also, we apply it in AG data, where the cluster are composed by combination of time point and spatial location. So completely different than the cluster extent-based thresholding used in fMRI data normally. If she wanted to talk with me later, I'm really happy to understand if the method can be applied in other problem, different problem than fMRI.
So thank you so much. We're at 20 past. So next up is a break, and I'll just remind that the talk today has a preprint that they can go read, and I guess they can find your email to contact you further. So the next session will be a series of talks, and that starts at 30 past. So we can thank the speaker again. Here come the little applauses and hearts.
Thank you. Thank you.